So how do we define media? Media means it is the nutrient containing various composition of ingredients for the survival of your plant, cell, tissue or organ when they are cultured in vitro. So we are providing all the nutritional requirements for that particular plant so that it can grow under the in vitro condition. In vitro means, as I told you, in the laboratory. But you see that all plants will not grow at the same time. Okay, like or the same type of media what you are providing. Some of them will grow in solid media, some prefer liquid media. So there is lot of variations in which the plant grows. Some tissues grow in simple media containing only inorganic salts and carbon, but then some other tissues require a complex media. So it is up to us or up to the researchers to find out what type of media is required for my plant. Media in the sense, what type of nutrition I have to provide. Should I provide a solid media for it to grow, or should I provide it a semi-solid media, or should I provide liquid media, or should I provide a complex media to them, or simple media? So this should be done by the researchers who are doing the plant tissue culture by a trial and error experiment. So the nutritional composition promoting optimal growth of tissue under laboratory condition vary with the respect to the particular species. With particular species, your nutritional components vary when you're growing them in the laboratory. Therefore, your media composition should be formulated considering the requirement of that particular species, and we have to provide it with the media to that plant. So what are the medias which is required? For example, like when we were talking about the history of plant tissue culture, we came across a word called White's media. So this White's media is particularly, I told you, it is used for growing your root culture. Similarly, I told MS media. What is the function of MS media? Your MS media, I told you, it is a hallmark media. Even today it is used so. So it induces organogenesis and regeneration of plant. Organogenesis means the formation of organ, because we are using simple cells, simple tissues, and sometimes even the organs. And we want, or your callus you're using, we wanted to develop all these organs, develop the cell division it should undergo. So probably we use our MS media. Similarly, for example, we have this B5 media. Okay, so this is another example like B5 media, which is particularly used for suspension culture. So there are many types of medias which are available, okay, of which MS media is important for you all from their point of examination. So now we shall look into the functions of the media. What all is required for the preparation of the media, or the ingredients what you require. First, we require the inorganic nutrients. Okay, your inorganic nutrients can be macro and micronutrients. We shall explain this all in the next slides. We have to provide water if it is essential. Minerals is required. We should provide vitamins and amino acids. The most important, we have to provide growth regulators. We saw that Haberland has missed the growth regulator, to add growth regulator in his differentiated cells, and we saw that it not divide. So growth regulators means it is the hormones, growth promoting hormones for the plant, that is required. And sometimes gaseous exchange is also required. That is, when you're preparing this suspension culture, we shall focus on the gaseous exchange there. And you should also have a provision to remove the plant metabolic waste. That is, plant also do produce some waste when they're growing in vitro under laboratory condition. You have to remove that waste. If you are not removing that waste, it can kill your culture. Okay, so that should be removed. Now, each of this we will study taking into separate subdivisions. So, according to this International Association of Plant Physiology, okay, what they have told is, for a macronutrient, the unit is millimole per liter. That is, you should use millimole per liter of whatever substance, whatever ingredients you are using for macronutrients. And for organic nutrients and for, micronutrients and hormones and vitamins, you calculate under micromole per liter. So macro means, it is more. So that is what it is, millimole per liter. And micronutrients, hormones and vitamins are required in small amount. That is why we weigh it in under micromole per liter. We do the calculation based on that. So the elements required in the concentration, if it is a concentration more than 0.5 millimole per liter, then it is called as macronutrient. And if it is less, okay, then 0.05 millimole per liter, it is a micronutrient. Otherwise, you can convert this into this millimole per liter, you can convert it into micromole also. So this is the concentration required, or the unit in which whatever you're weighing is represented. Okay. For example, you weigh it in grams per liter, milligram per liter. But here we use this terminology millimole, micromole per liter. Now first coming to the inorganic nutrients. So what did I tell? Your inorganic nutrient, it consists of macro and micronutrients. So macronutrients means immediately what flashes to your mind, it is required in larger quantity. So what are my inorganic nutrients are? It can be your nitrogen, okay, or it can be your phosphorus, potassium, calcium, magnesium and sulfur. These forms a macronutrient. Now all these are essential to your plant. Suppose say see, if my plant require nitrogen, I cannot add the nitrogen gas into my plant. Okay, I should use and substitute for it. For example, I can use ammonium nitrate. Okay, I'll write it in. I can use ammonium nitrate, or I can use potassium nitrate, or even I can use potassium dihydrogen phosphate. Okay, so these all I can use it. So what is happening? I'm providing a mixture of salt to my plant. So when I'm adding ammonium nitrate and I put it into water, what will happen? It gets disassociated. There is dissociation and ionization. So what happens is, for your plant it will become, or for your culture it will become easy to observe. So when it disassociates into ammonia and nitrate, what happens? It can easily get the nitrogen from here. It can get easily your potassium from here. So like this, we use a chemicals or mixture of chemicals from which your plant or your culture can derive the nutrients. So why is this nitrogen very essential? See, nitrogen, what it does, it influences your plant growth rate. It is a part of your DNA, part of your protein, chlorophyll. Okay, it is present in all the amino acids and hormones. Similarly, you see your phosphorus, it is present abundantly in your meristematic tissue. Meristematic tissues are what? They are the fastest growing tissue, and also the phosphorus is essential for the photosynthesis and respiration. When we come to potassium, you can see that potassium is essential for the cell division of our culture. Also, it is required for a meristematic tissue, and you can see that it helps in many of the pathways like your protein, carbohydrate, and chlorophylls. Similarly, you can see your calcium. Okay, it is involved in the cell wall and root and leaf formation, and also it is involved in other functions like translocation of your sugars, amino acid, and oxaloacetic acid. Magnesium is another macronutrient which is required for the plant. It is again involved in the photosynthesis and the respiration system, and also it is involved in the translocation of the phosphate and starches wherever it is required. And then lastly, the sulfur. You know that the sulfur is present in the amino acids. It is a part of amino acids and enzymes, and also it helps in the formation of nodules. So these are, okay, your calcium, magnesium, sulfur, phosphorus, potassium, nitrogen. These are all macronutrients which comes under inorganic compound, which are must and you should provide to the plant for their survival, because you have seen that these are required for various metabolic activity and to perform various functions of the plant. So the most important of macronutrients, like when we say, we tell it is nitrogen. Okay. So nitrogen, you can usually add it in the concentration of 2 to 20 millimole per liter. Otherwise, I can provide nitrate or I can provide ammonia. But the problem, you can see I have put an asterisk mark here. Okay. This ammonia has a problem because it is poisonous. In case of ammonia, only you're using ammonia, there is need to add one or more tricarboxylic acid, citrate or malate, so that any deleterious effect caused by this ammonia, okay, can be neutralized. So when nitrate and ammonia ions are present together in the culture media, you see that this is used up. Hopefully you understood. Once again, I will tell, whenever in a media you have nitrate and ammonia present, usually your cultures, they take up the nitrate and not much of the ammonia. But suppose say you are adding something ammonia salt, you shouldn't use ammonia salt alone. Why? Because your ammonia, when you're using it as alone, it is poisonous to the plant. That is why to neutralize its poison, okay, we use certain like this tricarboxylic acid or citrate or succinate or malate is added. So, and other what all macronutrients we are using, no, you can use it at a concentration 1 to 3 millimole per liter. Now coming to your micronutrients. So micro means you all know that it is in small quantity, okay? These inorganic elements are required in small quantity but essential for the plant cell and tissue growth. Though they are required in trace amount, they are very essential for your plant growth. And these include your iron, manganese, zinc, boron, copper, molybdenum. All this can be used as your micronutrient. And look at the concentration what you're using. It is quite less when compared to your macronutrient. So of all this micronutrient, you see that the iron seems to be more critical, more important. Why? Because it is in the chelated form of iron. We don't use simply iron as such. We use it in the form of chelated form, like it is in the combination with sodium EDTA or only EDTA. Why? Means this iron, what happens is, when you're using, we can't use iron directly. We are using it in the form of iron tartrate and citrate. This iron citrate or tartrate, when you're dissolving in the media, it starts to precipitate. Okay. Once it starts to precipitate, what happens? If it in the media, see if it starts to precipitate, what happens? They are not soluble. So your plant cannot consume that because it has already precipitated. So that is why usually we use this EDTA. Okay, iron with EDTA. So EDTA is what? It acts as a chelator. So what it does, what is the function of the chelators? So it has the ability to form complex bond with your metal. See here, this is my chelator, that is your EDTA. Now when you have added along with iron, what happens? They form a complex. Okay, once if they form a complex with your metal ion, so that your metal ions will remain in the solution despite of precipitating agents. So your plants can utilize them. Once again, I will tell you all, if I'm going to use iron tartrate or iron citrate, these iron will start to precipitate and they will not be available for your culture to grow because they are precipitated. Therefore, we will use certain chelators like EDTA, which forms a chemical bond with your iron. When such type of bond is formed, okay, here you see sodium salt of EDTA, they act as a sequencer or they act as a chelator. That is what happens is, even though there are certain precipitating salt in your media, okay, your iron will not precipitate. So it is easily available for your plant as a nutrient. They can take up it as a nutrient. So it is easy for the plant to utilize them instead of using this precipitate. Again, the other chemical what we use is the manganese. It is involved in the regulation of enzymes, growth hormones, and even it helps in the photosynthesis and respiration. So this is the importance of your iron. We use it in this form. So these are other micronutrients as earlier I mentioned, and you can use it in very small quantity. Next, coming to the carbon and energy source. So the most preferred carbon source for the plant is sucrose. Okay, sucrose is very easily available for the plant to utilize. After sucrose, it is the glucose which supports equally the plant growth, while fructose is less efficient. Okay, sucrose is the main carbon source what is used. Equally, even your glucose can be used and fructose can be used, but it is not as efficient as sucrose and glucose. So one important point what some of the scientists have noticed is when you're autoclaving sucrose, okay, when we're sterilizing this sucrose, what happens is it is converted into glucose and fructose. So when I'm autoclaving this sucrose, whenever I autoclave this sucrose, what has they have observed is this sucrose gets converted into glucose, okay, and fructose. So when I'm adding such type of autoclaved sucrose to the media, it has been observed that the cultures immediately take up the glucose first, indicating that glucose can be easily absorbed by the culture plant than your fructose. This is an important point what they have noted. So the first glucose is used and then they utilize the fructose. When all the glucose are finished in their medium, only then they use the fructose. Other carbohydrates, whatever we have like lactose, galactose, maltose, okay, these are not used. Why? Because they have provided inferior results. They have not given us any beneficial results. That is, therefore, we are not going to use these type of carbohydrates. So what is this? From where I'm getting my carbon and energy? How we humans get our energy? First source is your carbohydrates, right? Similarly, for your plant also, the important source of carbon and energy is coming from the carbohydrate. And which are the carbohydrate? It is the sucrose and glucose and then fructose, okay, to lesser amount. Whereas the others what we mentioned, your maltose, your lactose or galactose, raffinose, these do not provide any good results. And another important, when we are speaking about the carbon and energy, we know that all our plants are photosynthetic. That is in the field. But what about in my laboratory? The cells, tissues, okay, they do not have chlorophyll. So they are not autotrophs. That is, they cannot synthesize their own food. Sometimes you will see some green color growing in your plant, that the plant which is growing in your laboratory. So you may think that, okay, that green color is what chlorophyll and it has an ability to undergo photosynthesis by itself. No, that is not the case. Your tissue culture plants are not autotrophs. Okay, so that you have to provide carbon and energy to them. Only after the plantlet starts developing, after it has gone cell division, when it has started the cell division and when it's starting to produce different tissues or organs, only then it will try to become autotrophic. Otherwise, they are not autotrophic. Therefore, it is up to us to provide all the carbon sources. Now we have spoken about our inorganic components. Then we spoke about the carbon and energy sources. Okay. Then we'll be speaking about the organic supplement. So organic supplement, the first point is the vitamins. In vitamins, what happens is your plant synthesizes vitamins endogenously. That is, within itself, it can, by itself, it can produce the vitamins endogenously. And these are used as a catalysis for various metabolic functions. Similarly, in our body, how this vitamins we consume, it acts as a coenzyme and for the continuation of metabolic processes. Similarly, the plants also produces vitamins endogenously for various metabolic processes. But when plant cells and tissues are grown in vitro, whenever you are growing them in vitro, okay, when you're growing them under vitro condition, you must provide the vitamins. Though it is required in trace amount also, vitamins are required for us also in trace amount. But just imagine if we are deprived from vitamins, what happens? So many diseases can happen to us. Like if vitamin A is not present, blindness is, okay, thiamine is not present, beriberi. So though it is required in trace amount, it is very essential for the development of the plant, particularly the plant which is produced, which is grown in vitro. So when plant cells and tissues are grown in vitro, some essential vitamins are synthesized. They do synthesize, but only in suboptimal quantity. Okay, they do not produce it up to the maximum level. They produce it only to the suboptimal quantity. So hence it is necessary to supplement the media with the required vitamins and amino acids also. So we supply the thiamine, nicotinic acid, pyridoxine or calcium pentathenate, all this myoinositol as we have mentioned here. So we provide it. So once again, let us come back. We have told that vitamins are produced endogenously, but in the plant in vitro also they start producing. That is, when we are growing in laboratory condition also they produce vitamins, but it is very in suboptimal condition, which is not sufficient to proceed with their metabolic function. So therefore we provide the vitamins for the plant. And as I mentioned here, these are some of the vitamins which we supplement to the plant. And we can see that these are essential for all their metabolic activity. The essential has a coenzyme for citric acid cycle. And here you can see that it is part of cell membrane. So these are all required for the plant group. So coming to amino acids. Okay, amino acids also are produced by the plant, but even these are not in sufficient amount. They are not being produced. So your cultured tissue are normally capable of synthesizing amino acid necessary for various metabolic processes. But in spite of this, the addition of amino acid to the media becomes important. Why? Because we have to stimulate the cell growth, particularly your protoplast culture, and for establishing cell lines. For that and all, amino acid is required. Whatever is synthesized by the culture is not sufficient. So we have to add the amino acids. So you can see here the most common source of organic nitrogen used in the culture media are the amino acids. So which are the common ones we use? We use casein, hydroxylate, we can use glutamine, asparagine and adenine. So one important, very important point to what you have to notice is when these amino acids are added alone, okay, when you add individual amino acid to the media, they can be inhibitory to the cell. They do not allow the culture to grow when you are adding single amino acid. This is being noticed. Therefore, it is always essential for you all to take precaution. Instead of adding single amino acid, you add mixture of amino acid to your culture, plant culture. Okay, if this is your media and if this is your culture growing, you add a mixture of amino acids into it so that it can grow. Otherwise, it is proven to show that it has the inhibitory effect. Tyrosine has been used to stimulate morphogenesis, that is the different phases of the cell cultures. So also it is noticed that you cannot use tyrosine alone. Okay, it should be used in agar media. Only when it is used in the agar media, it becomes active. See, some of the reasons why this happens, it is still not known. But what happens is when we are adding this individually, we see that your plants do not grow properly. That is why we add it in the form of the mixture. So supplementation of culture media with adenine sulfate can stimulate cell growth and greatly enhance the shoot formation. So whenever we are using this adenine sulfate, okay, see adenine in the form of, we are using it in the form of adenine sulfate, it is very good for the development of the formation of the shoot. So here the most important point in amino acid what you have to consider is the plants require, if you remember, even White, when he did his experiments, he used yeast extract, and later on what did he do? He shifted to B complex vitamins, and he saw that if we use these thymine and all, it is not necessary for us to use yeast. So he showed us the importance of vitamins or these amino acids. Okay, so remember, you should not add single amino acid. Instead of that, you add the mixture of amino acids, so you get the best output from the plant. Now lastly, so these all are, I've used now inorganic, organic, we have used vitamins, amino acid, and now we'll see with some of the natural organic supplement. Okay, this also I told you when the history of tissue culture, certain scientists started using coconut milk, and they found that its property was very much different from the auxin. Auxin is of what? Plant hormone. Okay, so we can use this coconut milk, casein hydrolysate, protein hydrolysate, tomato juice, yeast. Many of this are being used. So what they have seen is that like even you can use your orange juices, tomato juices. So in tissue culture, the success and achievement with the use of coconut milk and protein casein hydrolysate has been significant. So when you're using coconut milk and casein hydrolysate, it has shown that these have positive effect on your culture, whichever you're growing, that is your tissue culture. Similarly, potato extract has been found to be suitable media for anther culture. I've not mentioned potato here. So even this potato cultures. So most of you have used this potato dextrose media. Okay, this potato culture media is also used for the culturing of anther. So generally the use of this natural extract is avoided, though you see that they have beneficial effect. Like coconut milk or casein hydrolase or potato, we saw that they have the positive effect on the culture. Usually these should be avoided in this tissue culture. Why? Means because we don't know the quality and quantity of this growth promoting. Okay, because we are all measuring and adding, but here we don't know the quality of it, from where it is derived. We don't know what is the quantity of content what are present in the coconut milk or your fish emulsion. So probably because of these reasons, it is particularly avoided. And therefore now what the scientists are doing is, like, instead of using tomato juice, for example, it is replaced with asparagine, your amino acid. Okay, now we see that yeast and malt extract, it is being replaced by asparagine. So instead of me using tomato juice or yeast extract or malt extract, I can weigh this asparagine because I know the quality of it, I know the quantity of it. I can use it instead of using these natural complexes. But still we see that some labs still using this coconut milk and casein hydroxylate and potato cultures. Okay, here it is, potato extract for anther culture. Lastly, we shall talk about the charcoal, activated charcoal. You can see in the box I've written a paradox. Okay, why? We shall see that. See, the addition of activated charcoal to your culture media is reported to stimulate the growth and differentiation in some plant. Okay, it's when you add this activated charcoal, okay, you can see its positive effect. That is, it is stimulating the growth and differentiation of your orchid plant or carrot or tomato. But the same activated charcoal, when it is added to soybean or tobacco, it has shown negative effect. That is why it is a paradox. Either it can activate or it can act as an inhibitor. Now, why this is happening? So why charcoal is doing this? Because this activated charcoal, okay, here for this plant, wherein it is acting as an activator, what it is doing is, it is trying to remove all the waste from that culture. So what it is doing, when the waste products, waste metabolites from this is being removed, it is actually enhancing their growth. That is why. Whereas why here it is acting as an inhibitor means, when we are adding activated charcoal here, it is showing negative effect. What it is doing is, it is taking off the hormones, the phytohormones, which are essential for the growth. Okay, that is what the scientists have observed. So it can have beneficial effect, it can have this negative, not beneficial effect also. When it is having its benefit effect, we see that it is trying to remove the impurities in the culture. On the other hand, here what it is doing, it is not removing the waste from the plant culture, but instead of this, it's removing the hormones itself from the culture media. So suppose say this is your culture media and you're growing your culture here, okay, and I have added, this is your soya bean plant or tobacco. When I'm adding the activated charcoal, all your nutrients are present here. What it is doing, it is absorbing of your nutrients itself. Then where is the chance for the plant to grow? It will not grow. Okay, so it is having its merits and demerits, but particularly remember, activated charcoal is used for detoxifying the waste from the plant. So whenever you are using this activated charcoal, first you have to see that it is acid washed. It should be neutralized, because we are using acid, it should be neutralized with a base, and then if it is necessary, only then you use your charcoal.